I'm not going to spend a lot of time. This is a disease, and there is nothing more frightening to a pharmaceutical executive or to a patient to be taking a drug for a benign disease and have this disorder, which is torsade de pointe, and 1 out of 5 times you're going to die, and 5 out of 5 times you're dead. So the history goes like this. This was a matter of great concern. I'll go into a little more detail about it. There have been some regulatory guidances. Those have been usurped more recently, and a new approach, which you'll hear me talk about, is called the Comprehensive Proarrhythmia Assay, or CIPA. And what work that we have done on this, work that we think is important for going further, which we call quantitative profiling, but sort of a modified version of CIPA. And at the end, probably won't spend enough time on this, but the cost benefits of a proper approach in vitro are humongous, and it's very poorly appreciated by drug developers. So the history looks like this. Torsade de pointe, it's a polymorphic ventricular tachycardia. It's always associated with QT prolongation. But the flip side isn't true. QT prolongation is not always associated with torsade. So it's a false positive issue in the main as far as we know. We don't know about the false negatives because we often don't hear about them for reasons of competition, et cetera, et cetera. So when this whole thing started, it looks like there was an epidemic. There were at least 7 drugs that were withdrawn between 1999 and 2003 from a variety of classes. I can't go into that right now. So the regulators, via the International Conference on Harmonization, were concerned about QT prolongation because the frequency of torsade de pointe for non-antiarrhythmic drugs is incredibly low. It's on the order of 1 in 50,000 to 1 in 100,000. You cannot do a clinical trial to pick this out. So they chose a surrogate. And the surrogate marker that they chose was QT because, as I told you, TDP has QT, but QT doesn't have TDP. So that was the problem from the get-go. And so, as it turned out, there were discordances. For example, drugs like verapamil, phenobarbital, pentobarb, ranolazine more recently, are all drugs that are hERG blockers. Which was so the workup was really hERG and then an animal QT, and so drugs that were hERG blockers were discounted. And what happened actually in the industry was because the industry is highly risk averse. You can't blame them for that. It costs a ton of money to make a drug, and the idea at that time was that if it had a whiff of hERG, it was all over for that chemotype or whatever. And everybody was aware of this. The FDA, Congress, et cetera. The pipeline sucked. I mean, there was more and more money plowed into this stuff, but the number of new molecular entities that came out at the end of the trail was getting smaller and smaller. From the FDA's point of view, the process had been perverted, and it was pretty clear that you needed alternatives for this lousy surrogate. QT prolongation, I mean, if I yelled at you, your QT would change way more than the QT indications that are used to determine whether a drug has a signal. In other words, the QT can vary during a day by 75 milliseconds. The upper bound of the 95% confidence limit in a thorough QT trial in the clinic is 10 milliseconds. So, it just was sort of, the whole process was screwed. So they decided to come up with proposals, and this is this comprehensive thing in which we've been quite heavily involved, and which looks at things in a little bit different way, and I'll show you the reasoning for why we want to do it that way. So just very, very quickly, here's the arrhythmia. Is generally, you're taking the drug, you have a short, long, pause, short, and then boom. There's the arrhythmia. And this has to do with the vulnerable period and the next beat coming right in on the T wave. Remember that in cardiac propagation is active. Propagation of depolarization is active. but repolarization is local. And that's why all the trouble is during repolarization, because you can get asymmetries in the wall. And so basically the idea was to shift from looking at QT and start to look for these signals of proarrhythmia or proarrhythmias themselves. I don't have time to go into the reentry. There are various models for what goes on with this stuff. Oh. So anyway, so what are the major players here? The major players are obviously the inward currents, which are the sodium current and the calcium current. And what's not shown here is that there's a very slowly inactivating component of the sodium current that contributes to the plateau of the action potential. Remember, during the plateau phase of the cardiac action potential, the membrane resistance is actually greater than it is at rest. So very tiny currents can have very big effects on the potential. So you've got a little current in here, which we'll refer to later. It's called the late current. This is the peak current. And then the calcium current looks like this. And then the rapidly repolarizing current, which is heard, looks like this. It's a really, really great current, but it's just wonderful design. And then there's the regular delayed rectifier, and then there's an inward rectifier. These are really the main players. You can add stuff to this, and people are adding stuff to this. But right now and basically I'm going to be talking about this guy, and this guy. Okay, so what is SIPA all about? SIPA says, okay, we're not looking at the QT. We're not looking at action potential propagation. We are going to look at 4 buckets. We're going to look at the functional effects of the drug on multiple cardiac currents. So these are what I call multiple ion channel effects, or MICE. We're going to do it automated or manual. Manual is kind of out the window, because if you're doing 3, it's very easy to do the arithmetic, but if you do 3 channels and you do them manually, it's going to take you God knows how many days and how much money. At $1 million a day, it turns out to be $100 million if you do it that way, just for 3 channels. Whereas if we use the Q-patch HT single hole, because it gave us gigacil resistances, and we were very careful about how we used it. But you can do all this stuff in 3 days. So it starts to get to be very important. Then we did something called safety assessment. I'll talk about all that. And we've always, since we are looking at, in the proarrhythmia game, we're looking at whether a patient has torsade or does not have torsade with a drug, okay? So it's a dichotomous arrangement, which is really pretty favorable for a logistic regression type of analysis. And that's the way we approached it. So this is some of this is a little unique to us. Then SIPA says, well, okay, so what you should do is take models of left ventricular action potentials and plug the numbers you get for the IC50s into those. So basically, if you've seen, the Hodgkin-Huxley equation, which can be converted into a Markovian model, basically what we do is we simply scale the G-bars for the different currents by the IC50s. Now, you can get way more involved if you want to do on-off kinetics, but I think that's really something far downstream in terms of the process. Then the idea is to try to experimentally confirm this, generally using induced pluripotent stem cell-derived cardiomyocytes, human of course, and something that is used as an electrophysiology surrogate is multi-electrode array. I don't really have time to get into all of that. And so then you come up with some mechanism-based conclusion. But this mechanism-based stuff is really pretty vague. It's a continuous scale, but if you got all these guys that you're looking at, how do you weight them? And we don't know yet at this point in time exactly how to weight the different components. So that's going to be very, very important. And so then you will make rank-ordered comparison about the various drugs that you're looking at. And then there's the contextual data. So we're dealing with the effects of drugs on normal tissue. But people without a disorder don't take drugs usually. I mean, I'll leave that. That's questionable. But anyway, if you're taking a drug for, let's say, even a benign disease, or if you're taking a drug for potentially dangerous disease, it's not enough to have considered what this scheme's talking about, because this is referring to normal tissue. You have to put it in the context of the disease that you're trying to treat. And that includes looking at comorbidities in particular, okay, which come in all different categories. So this is really somewhat undefined at the moment. So all of the work I'm going to describe about has been in 2 papers that have appeared in scientific reports in 2013 and 2015. Basically, when we did this stuff, we got 55 drugs. 32 were tersatogenic and 23 were not tersatogenic. That's not really the way the world runs, but that's what we did. And there's an outfit called Credible Meds, which tracks these drugs constantly, has been in it since the early days. We primarily use Credible Meds for deciding whether drugs were or were not tersatogenic. And then we used other things like literature references and in particular the package inserts and whatever stuff we could squeeze out of FDA, which is usually very, very little. So when we looked at the IC50 for HERG, for example, which was one of the ones we looked at very closely, here's our data here against the different drugs, and here's the data from the literature. So it looked pretty good, automatic versus manual, manual being the reference standard. So then we actually took 29 of our subset and did, again, the Q-patch HT single hole against manual patch. And here's the line of identity, and here are the 3 SDs from that line of identity. You can see that there's a couple of points that fall off, and this has to do with the hydrophobicity of the drug. And actually, you can minimize this if you're aware of it, because you can prime the system in advance to minimize this. But nevertheless, that is something that you have to be aware of, because the system is a carrier with glass at the bottom, and so the carrier can interfere with the amount of real drug that's being exposed. So then we went in and did IC50s on HERG against these drugs, on CAV 1.2, which is the main cardiac calcium channel, and on NAV 1.5, which is the main sodium channel. And these are the circles represent the means and the 95% confidence limits. So the data are very, very tight with this system. There's an occasional problem which has to do with solubility effects, but as I say, that can be managed. All right, so what do we get? So 2 of the drugs, one of the drugs that was really kind of a poster drug for the issue was verapamil. Verapamil is a drug that is still in the clinic, but it would never have made it because it's a HERG positive drug. However, it's also a calcium channel blocker. Terfenadine, which was the poster drug, which got me into this business actually, is a drug that is widely used, but is a pretty selective HERG blocker. So if you look at the ion channel IC50 for these 2 drugs, and you look at the values for HERG, which is the red deal, and NAV 1.5 and CAV 1.2, and then you look at the exposure level in the blood as determined by the Cmax and Tmax, you can see that for this guy, these are widely separated. Okay, so in other words, the exposure level is greater than the safety margin. On the other hand, if you look at verapamil, you can see that it doesn't do much to sodium, but the exposure level and the calcium channel block and the HERG block are similar. So you have the outward block, the block of the outward HERG current, and the block of the inward calcium current neutralizing each other. And that's what makes this drug safe. So then we came up with this sort of way of looking at it. So now we're plotting the IC50 over the effective therapeutic plasma concentration. So this is the safety margin kind of deal. This is the reference line for whether a drug is going to be tersatogenic or not. Below this level, it should be tersatogenic based on HERG block, and above this level, it should not be. And you can see that we got these V plots. So here's terfenadine, which is a potent HERG blocker. It does very little to CAV or NAV. versus verapamil, which is equipotent or perhaps even more potent as a calcium channel blocker than as a hERG blocker. So we use these kind of V curves to identify the drugs of interest. And now over here, what you can see is that, you know, a lot of the torsatogenic drugs have these upright V curves, even if they're in this gray zone between 10, a safety factor of 30, and a safety factor of 100. On the other hand, look at these guys in here. These guys in here would be tossed out, right? I mean, they're clearly below this safety margin level, but in fact, they're not torsatogenic. So they're kind of inverted V curves, and that's what we refer to as safety curves. They're V curves that we look at very quickly to figure out where we are in the process. Okay, so how do we analyze the data? So since we're using logistic regression, the expression for the probabilistic expression for this is indicated here for, let's say, the case of hERG, and here's for being torsatogenic, and here's the probabilistic expression for a model in which hERG and calcium IC50s are being considered. So if we lump the drugs into groups of 10, we can look at, for example, the frequency of torsatogenicity with a drug that becomes more and more potent as a hERG blocker, and you can see that the raw data follows these filled circles. And if we look at a model, and we call this one Model 1, which just looked at hERG alone. Now, if you're looking at hERG and CAV and NAV, and two forms of NAV, you can have lots of models, okay? But at the end of the day, the models that worked for us were a model that were the best to compare were the Model 1, obviously, and a model in which you considered CAV and hERG. And I would think now, because of our interest in this late sodium current, we're going to probably have to expand that model, but we haven't at the time, and here's where we were. So it turned out when you do the expression for CAV, you actually don't even need to know the effective therapeutic plasma concentration. You simply have to know the ratio of the IC50s. And so when we looked at, and we called this model, as it turned out to be the best of all the models, you can see that the raw data looks like this, look like this. And these are the drugs that we made mistakes on, outlined in the red. So what we did was to fit the raw data. And how do we get the raw data? So the raw data came from this training set of 55 drugs. So how did we get a test set? We got a test set by leaving out one of those 55 drugs, one at a time, and recalculating the values for that particular drug that fit into these kind of expressions here, okay? So then we had a test set, and this leave one out is a customary way of doing this when you have very limited test sets available to you. So when we fit the cross-validated data, this was the kind of fit we got, and this was the kind of fit we got here. So you can see that with the hERG, we're looking at tenfold groupings of 10 at a time, and you can see with the Model 1, there's not that great differentiation, but with Model 5, there's clear differentiation between the drugs that are torsatogenic and the drugs that are not torsatogenic. And a very nice way of looking at this is to plot the sensitivity of your drug, that is to say the true positives, against 1 minus the specificity of your drugs, which are the false positives. And you generate curves based on the sort of cutoff frequencies that you use to segregate the non-torsatogenics from the torsatogenics. And actually, when you do it this way, these are called Roc Curves, which is an acronym for Receiver Operating Characteristic Curves, very widely used, both in evidence-based medicine and was discovered actually by the British, by British radar for identifying submarines back at the start of the Second World War. So which ones were Germans and which ones were ours? So, oh, wow. Okay, well, I got to go a little faster. Anyway, the very important point here is to get the same number of true positives, you have to have more than twice as many false positives with Model 1 as you do with Model 5. So that justifies the process. And you can see that with Model 1, you had 6 false negatives and 8 false positives. With Model 5, you greatly diminished this. If you go through all of this now and you look at the test properties, this is the positive predictive value. How likely are you to use the assay and get the prediction of torsade? How likely are you to use the assay and not get torsade? And you can see that the odds increase tremendously if you use the mice assay. And the likelihood ratio model looks at it the other way around. How likely, if you have the disease, how likely is your test to be positive or negative? And once again, it turns out that you get a very, very significant, a huge improvement actually in odds with this approach. So just very quickly now, quantitative profiling. Bottom line, there's a lot of stuff over here. Bottom line is we think that you have to do this stuff. If you're going to look at multiple channels, you should use the same protocol for all of the channels. That is, the frequency of pacing should be the same. The ramp stimulus should be the same. It should simulate a ventricular action potential to the best you can, and apply that to all the different channels. And I won't go through the data over there, except to say that what we found, as you might expect, when we looked at the safety margin for verapamil, we got what we expected. When we looked at, we got the V curve for dofetilide, which is a selective hERG blocker. We got what we expected. And then there were 2 drugs we were kind of interested in, vanoxaprine and bepredil, which gave the same safety margin profiles, and yet bepredil is a torsadogenic drug, and vanoxaprine, based on the criteria we used, was not a torsadogenic drug. And so the question came up as, well, how's this? How's this happening? And we knew from experiments we'd done 10 years ago using trafficking, which a process that we had developed in the lab and we're very familiar with, that bepredil gave a trafficking signal. So we went in there and well, let me just back up very, very quickly. With the data that I showed you, the mice data, we could simulate the action potentials in all these different cases. And this is the experimental data actually using the stem cell-derived cardiomyocytes. But the model data looked very much the same. You see the markers for dofetilide, and you don't see them for these guys. They obviously don't have time, right, Andy? So we thought that trafficking might be an issue. Trafficking you've heard a little bit about. You put a tag in there, do some chemiluminescence. You can also track the proteins, and you show that there's the image densities are dose dependent, as well as the current you see. Now you have to incubate overnight because the lifetime of the hERG channel is roughly 16 to 24 hours. So we went in there then and tried to figure out what the differences were, and we looked at the relative surface expression because we're actually counting. We are counting with this method the number of channels that are at the surface, all right? And we can actually lyse the membrane and count the number of channels that are there. And so at the end of the day, it turned out that bepredil was a clear trafficking inhibitor over the relevant concentration range, whereas vanoxaprine was not. There's something in that curve that some sharp guy in the audience might pick up on, but I'll move on from there. So we've actually added another box, which is that you should use trafficking and more chronic exposure. I think that's a given. We've heard talks yesterday about this sort of thing, and I think we need to using automated patch clamp. I think we probably need to pay a lot more attention to this. Factors, same story as before. Weighting, very, very important. Context, very, very important. Cost benefits, last slide. Drives me crazy why there is so much resistance to doing the right kind of stuff early on. So if you look at the costs in early discovery and late discovery, and then of course phases 1 to 3, and let's say you do the full bore. If you do HERG, you're looking at 20,000. If you do the full bore thing, you're looking at 60 to 80. Maybe now you start adding some, you're doing 100,000. So what happens if you stop progressing the drug? Well, by this time, this is small compared to this, so you're going to save hundreds of millions of dollars, if not close to $1 billion. And what if you say, okay, you know what? I'm not the usual risk-averse guy that runs pharmaceutical companies. I'm a guy that's going to stick my neck out because I've got pride. I'm going to stick my neck out and try to take this drug across the finish line. That guy takes the drug across the finish line, and he's going to make billions for the company. Will he be rewarded? I don't know. Yeah, somebody who's familiar with the industry has to tell me, but I doubt it. I think the safety guy covers his butt, and the risk-taking guy may or may not get what he really deserves, which is some kind of medal in dollars. Okay, so here we go. Compared to this, the mice assay improves the logistic regression model, Roc Curves, and the predictive values, both positive and negative. We have validated a medium throughput automated patch clamp, very important. It's likely that these targets will be extended. FDA is talking about these 3 guys. We think quantitative profiling is the way to go. However you do it the same way at something that reasonably approximates the way the left ventricle fibers work. I think exposure durations are important for lots of reasons. The weighting and the contextual considerations I've spoken to, and we can show that CIPA will reduce drug development costs almost by $1 million. So here are the guys that really did the work: Jimmy Kramer, Carlos Obejero Paz, Andrew Bruning-Wright, and Peter Harlick. Thank you.
Thank you both. I think we have time for probably just one question.
So very nice. So where do you see the limit for these in vitro analysis? Can we totally leave out animal experimentation?
No. No. At the end of the day, you have to do that. But the question is, when you do it, and with how much information are you forearmed? So yeah, there's no doubt about that. The CIPA is very clear on this point that the animal stuff is going to have to be done before it goes into man. But the animal stuff is very, very tricky. It's just as tricky as doing a thorough QT trial, perhaps even more tricky because when you do the human trial, you can at least reason with the subject. And as a matter of fact, many of these companies that do thorough QTs have a group of trained humans, who don't get upset when the nurse comes in or whatever. You can't do that with animals. So, you have to do that. But I always think you're better off if you know what you're doing when you come to do it than if you don't know what you're doing when you come to do something.